Hi everybody. Now I am going to discuss about energy production by photosynthesis. Plants, algae and cyanobacteria are photoautotrophs. They have light as their source of energy and carbon dioxide as their sources of carbon. Carbon dioxide to be useful for metabolism, it must be reduced to carbohydrate. This process by which light is used to convert carbon dioxide to carbohydrate is called photosynthesis. Overall reactions: 2H2O plus CO2 in the presence of light, it gives carbohydrate plus O2 plus H2O. Photosynthesis has two important requirements. First is a large amount of energy in the form of ATP, and second one is a large quantity of a chemical reductant, in this case water. Several groups of bacteria, the photoautotropic green and purple bacteria, are also characterized by their ability to perform photosynthesis. But unlike plants, algae and cyanobacteria, they do not use water as their chemical reductant, nor do they produce oxygen as one of their end products of photosynthesis. Types of photosynthesis. There are two types of photosynthesis. One is cyclic photosynthesis, otherwise said to be is the anaxigenic photosynthesis, O2 not evolved, and second one is non-cyclic photosynthesis or axigenic, O2 evolved. First of all, cyclic photosynthesis. Cyclic photophosphorylation, as it is an axigenic photosynthetic bacteria. Anaxigenic photosynthetic bacteria possess chlorophylls called bacteriochlorophyls that differ from the chlorophylls of plants in structure and in light absorbing properties. Bacteria chlorophylls absorb light in the infrared region 725 to 1035 nanometer. This is the cyclic photosynthesis image. When bacteria chlorophyll absorb light, the energy of light raises the molecule to an excited state. In this excited state, an electron is donated by bacteriochlorophyll and it accepted by pyridoxine. That is, the electron carrying some of the energy absorbed from light is transferred to an ion containing heme protein is known as pyridoxine. From there it is passed successively to ubiquinone to cytochrome B and cytochrome F, and finally back to the positively charged bacteriochlorophyll. The energy released in the step between cytochrome B and cytochrome F is used for cyclic photophosphorylation, the generation of ATP from ADP, and second one is non-cyclic photosynthesis. Non-cyclic photophosphorylations or axigenic photosynthesis. In plant, algae and cyanobacteria, non-cyclic photophosphorylations will be occurs in photosynthesis. In this, light absorbed by pigment to photosystem, and this raises the molecule to an excited state and release an electron. This electron transfer to plastoquinone to cytochrome B to cytochrome F and finally to pigment 1 photosystem 1, and this image is non-cyclic photophosphorylation. Then last slide continuation is the ATP generation photophosphorylations occurs the step between cytochrome B and cytochrome F. When pigment system 1 absorb light release an electron, this electron is transferred from pyridoxine to flavoprotein to NADP+. Photophosphorylations occurs in the step again between system 1 and pyridoxine. The NADP is the final electron acceptor. Since the electron lost by pigment 1 is not cycled back, this process differs from cyclic photosynthesis. The donated electron from pigment system 2 is replaced. The breakdown of water is called photolysis and continues this process. The role of plastroquinone is the transport of proton on originating from water. This modified process has been termed exogenic photophosphorylation. Photophosphorylation, and this is the non-cyclic photophosphorylations. In this, light is absorbed by pigment 2 system, and this raises the molecule to an excited state and release an electron. This electron is transferred to plastroquinone to cytochrome B to cytochrome F and finally to pigment 1 photosystem. An ATP generation photophosphorylation will be occur in the step between cytochrome B and cytochrome F. When pigment system 1 absorb light release an electron, this electron is transferred from ferridoxine to flavoprotein to NADP+, and this is subsequently photophosphorylations will be occurs in the step again between system 1 and ferridoxine. The NADP is the final electron acceptor. Since the electron lost by pigment 1